Hello, dear students. Let's talk about one small topic, that is explanation of the concept of dominance. It is given in NCERT in a short paragraph, but sometimes you get a question from this part, so we need to understand this in proper manner. So, concept of dominance means actually we are using a term dominance, incomplete dominance, codominance. So, actually, what do you mean by a dominance? What is dominant allele? What is recessive allele? What is a dominant trait? What is a recessive trait? That can be understood here in a proper manner. So if we talk about this explanation of this concept of dominant, we know that every gene contains the information to express a particular trait. Any gene you take, we are using generally the term A, B, C like name we give. Sometimes we give a name like X, Y, Z. Whatever the name is given to the gene, that gene is has a information to express a particular trait. In a diploid organism, there are two copies of gene, and thus that is known as alleles, a pair of alleles. But in haploid, there is only one gene, one allele. But in diploid, there is a two allele on the homologous chromosome at the same locus. Now, if we talk about these two alleles that need not always be identical, means they can be identical, like capital T, small t, but that need not always be identical because we have a heterozygote condition like capital T, small t that we have discussed. So, if we take an example, suppose a gene that contains information for producing an enzyme. Suppose any enzyme is there, it is produced by a particular gene. Now there are two copies of this gene in a two allelic form. In this two allele, if we assume as in more common that the normal allele produces the normal enzyme. If when substrate is converted into the enzyme, if particular gene is producing an enzyme and there is a proteinaceous enzyme, the normal allele produces a normal enzyme. And it has a information for the what transformation of that particular substrate to product that enzyme has. So normal gene allele is producing a normal enzyme, but abnormal allele, modified allele is not able to produce a normal enzyme, or it can produce a normal enzyme as well. So what are first modified and unmodified allele that we have to understand? If we take a term, the unmodified allele, then it represents the original phenotype, unmodified, which is not changed, which is as original as it was found in the initial plants or initial organism. So it is a represent the original phenotype. So whichever the allele produce the original phenotype, that is a dominant allele. And the modified allele is generally the recessive allele. Dominant allele are usually more unmodified one, and recessive allele are usually modified. So we can say capital T was the unmodified, but some modification happened and it converted into the small t. So if it get modified, some mutation can occur, or due to any environmental factor, suppose the dominant allele is getting modified into the recessive allele, or original allele, or wild allele, or unmodified allele is getting modified into the modified allele. So the modified allele could be responsible for the production of. Means if modification happened in the dominant allele, this is not such that the phenotype get changed always, because the modified allele could be responsible for the production of normal or less efficient enzyme. The gene is modified, allele is modified, not it is all wild one or original one. But if it produces a normal or less efficient enzyme, then it can produce. It can also produce a non-functional enzyme. The modified allele will produce an enzyme, but it will be non-functional. It is not converting substrate into the product. And it is also possibility that the enzyme will not produce no enzyme at all. So if a modification occur in the original gene, then such type of possibilities are there, can be observed by the modified allele. In the first case, the modified allele is producing a normal or less sufficient enzyme. Means the modified allele is acting equivalent to the unmodified allele. So it will produce the same phenotype or trait, that is the result of transformation of substrate into the product, means the substrate will be product produced converted into the product same as it is converted by the dominant allele or unmodified allele. So here modified allele acting equivalent to the modified allele. Such equivalent allele pair are very common. But if the allele produces a less functional enzyme or a non-functional enzyme or a no enzyme, the phenotype is affected. Means in the second and third case, the phenotype is going to be affected or phenotype is going to be changed. So the phenotype or trait will only be dependent on the functionality of the unmodified allele. Either unmodified allele is functional or non-functional. It is changed much or not. That will define the phenotype. So let's see some of the questions which are not always be identical. The two alleles need not always be identical. If the allele produces a less functional enzyme, obviously the phenotype is going to be affected. Less functional enzymes may be affected. Okay, so I hope you understood this concept of dominance.